All right, got a big biology quiz on the horizon? Well, you've come to the right place. We are about to do a super fast tour of the Nobel Prize breakthroughs that literally built the world of modern biology. Think of this as your ultimate study guide.
So quick question for you. When you close your eyes and picture a living cell, what do you actually see? Is it just kind of a blob? Or do you see this bustling, microscopic city with little machines and messengers zipping all over the place?
You know that detailed picture of a city in your head? That's actually a pretty new thing. For most of scientific history, what went on inside a cell was a total black box. We were just guessing. But then a whole series of Nobel-winning discoveries came along and, well, they turned on the lights. So, how did we get from darkness to light? We're going to break it down into 5 key areas, perfect for your quiz. We'll start with how we learned to see and edit cells. Then, we'll look at how we mapped their machinery, harnessed our immune system, started building totally new biology, and finally, how AI is changing everything. Let's dive in.
Okay, first up, the most fundamental challenge in biology. You can't understand or fix what you can't see. This first section is all about the incredible tools that took us from just watching life happen to actually rewriting its code.
And believe it or not, it all began with a glowing jellyfish. The 2008 Nobel Prize went to the discovery of something called green fluorescent protein, or GFP. Just think of it like a tiny biological light switch that scientists can stick onto any protein they want to study. All of a sudden, they could actually watch molecules moving around inside a living cell in real time. This was an absolute game changer.
So GFP let us see that things were moving, but the picture was still pretty fuzzy. For over 100 years, a basic law of physics said there was a limit to how small we could see with a light microscope. Well, the 2014 Nobel for super-resolution microscopy basically shattered that limit. It's like going from seeing a blurry city skyline at night to being able to see individual cars on the streets. We could finally see molecules interacting up close.
Okay, so now we can see what's going on, but what if we want to change it? Well, that's where CRISPR-Cas9 comes in, the winner of the 2020 Nobel. And let me tell you, this wasn't just another step. This was a monumental leap. Scientists finally had a tool, like a pair of molecular scissors, that could be programmed to find one specific gene in a sea of 3 billion letters of DNA and then cut it, edit it, or even replace it.
So for your quiz, you really only need to remember 2 key parts. First, there's a guide RNA. Think of it like a biological GPS that you program to find an exact address on the DNA. Second, that guide is attached to the Cas9 enzyme. Those are the scissors. So the GPS takes the scissors to the exact right spot and, snip, the cut is made. It's just beautifully precise.
Knowing the genetic code is one thing, but that code is just the instruction manual for building proteins, the tiny machines that do almost all the work in our cells. To really understand how these machines work, we needed to see their exact 3D shapes. We needed their atomic blueprints.
Now, getting those blueprints was a really tough, long journey. Back in 2009, the prize went to the team that finally mapped out the ribosome, the cell's protein-making factory, using a technique called X-ray crystallography. Then in 2012, the same method helped us see the structure of crucial cell receptors that, get this, nearly half of all our drugs target. But the real revolution was still on its way.
The big breakthrough was cryo-electron microscopy, or cryo-EM. See, the old method required scientists to grow perfect tiny crystals of each protein, a process that could take years, if it even worked at all. Cryo-EM completely changed the game. Now, you just flash freeze the molecules in place and take their picture in atomic detail. No crystals needed. This massively accelerated drug development and even helped us design vaccines, like the one for the Zika virus.
So we've seen how we can look at and even change life's building blocks. Now let's turn to one of the most complex systems in our body, the immune system. The next set of discoveries taught us how to go from just understanding our body's defenses to actually engineering them into powerful medicines.
The 2018 Nobel recognized this really clever trick called phage display. It basically uses viruses that only infect bacteria as tiny factories to generate billions and billions of different human antibodies. Scientists can then sift through this massive library to find the perfect antibody to fight a specific disease, from arthritis to cancer. It's the technology behind some of the most powerful drugs on the market today.
But what about when our immune system goes haywire and starts attacking our own bodies?
Well, discoveries that led to the 2011 Nobel helped us understand how our immune system is activated. And a key part of that system is a type of cell called a regulatory T cell, or TREG. The best way to think about TREGs is as the immune system's peacekeepers. Their job is to tell the other immune cells to stand down, preventing them from causing autoimmune diseases. Understanding them is absolutely vital.
And of course, there's the 2023 Nobel, a discovery that affected literally everyone on the planet. The idea of using mRNA to deliver instructions to our cells wasn't new, but there was a big problem. Our immune system would see it as an invader and destroy it instantly. The breakthrough was figuring out that by making one tiny chemical tweak to mRNA's building blocks, they could make it invisible to our immune defenses. It could slip in, deliver the instructions, and train our immune system, the science that gave us the COVID-19 vaccines.
Okay, so far we've talked about tools for seeing and editing biology that already exists. But what if we could take it a step further? What if we could actually create brand new biology? What if we could take the most powerful creative force in the universe, evolution, and put it to work for us in a lab?
That's precisely what Frances Arnold did, and it won her a share of the 2018 Nobel. Her breakthrough is called directed evolution, and it's basically evolution on fast forward. You take a gene for a protein, you make millions of random mutations, and then you test them all to see which one is slightly better at a new job, like creating biofuels or manufacturing a drug. Then you take that winner and you do it again and again and again. It's an amazing way to create brand new proteins that nature simply never thought of.
And that brings us to our final chapter, where biology crashes headfirst into the incredible power of artificial intelligence. We're now entering an era where we're moving from just discovering the code of life to actually predicting it with computers.
But before we could run, we had to learn to walk. And the groundwork for this whole AI revolution was really laid back in 2013. That year's Nobel went to the scientists who built the first computer models that could realistically simulate molecules as they moved and reacted. It was like creating a virtual reality lab where you could watch chemistry happen. This was the critical first step. And what came next was a quantum leap. Just look at this. Before, figuring out the 3D shape of just one protein could be a student's entire PhD project. We're talking years of work. Then came an AI program called AlphaFold. And suddenly, we could predict the structures of hundreds of thousands of them with incredible accuracy in just a matter of days.
The 2024 Nobel recognized the teams that essentially solved what's known as the protein folding problem. Using deep learning, AlphaFold can look at the simple one-dimensional string of a protein's building blocks and predict its incredibly complex final 3D shape. This has been a grand challenge in biology for 50 years, and now it's been solved. It's completely transforming how we design drugs and understand the fundamentals of life itself.
And there you have it. From a simple, glowing jellyfish to an AI that can predict the machinery of life. Every single tool we've talked about today was pure science fiction not that long ago. So it leaves you with a question, right? With all these amazing new tools, what's the next breakthrough going to be? What will they make possible? Okay, now go ace that quiz.